and we're really honored to have them here to talk. So this is going to be the last NSF RCN webinar of this calendar year, and we're hoping that we can start up again in January. If anyone here has any suggestions for speakers, please reach out to me or any of the co-PIs, including Heseth, who is on the webinar as well today. The way the webinar will work is Marc will give a 20-minute talk followed by 10 minutes of Q&A, and then Jana will go second and give 20-minute talk and 10 minutes of Q&A. Everyone should put the Q&A questions into the Q&A chat, and from that window, you have the ability to upvote a question if you want to hear that question asked. I will be the moderator for the Q&A for Marc, and Heseth will be the moderator for Jana. If you put things in the chat, we won't necessarily see them, so please use the Q&A. So with that, we're going to go ahead and get started. So Marc, you can go ahead and share your screen, and I'll ask all the other panelists to mute themselves and stop sharing their video.
All right, we good to go? Can you hear me?
Yes, all good. Thanks.
All right, thanks so much for the invitation. It's really nice to talk to a group that, where I don't have to start off by explaining why wastewater can be useful. I'll assume that most of you are convinced now that this is kind of a cool thing to be doing for monitoring SARS-CoV-2. So in this talk, I'm mostly going to focus and not on the whole picture, but on the things where maybe we've done something that's different than what most people do, and some of our sort of interesting stories along the way and things we've been finding. To start with, in Missouri, this is actually what's really set us apart is it's a very equal collaboration between 3 different groups. It was actually kicked off by the Department of Natural Resources in Missouri in the state level, and they recruited my own lab and Chung Ho Lin's lab, who is my counterpart, and just whoops, where did his picture go? Dang it. He's over in he's basically a chemist that works on lots of environmental products, and also the Department of Health and Senior Services, which handles all of the communication, the websites and whatnot, and curation of the data. And we've got along the way picked up a bunch of other collaborators from other departments in the state. Now, first, just so the system we're using is good old-fashioned filtration and PEG precipitation. Same thing I've been using for 20 years as a virologist, which is what I did before I got sucked into wastewater testing. It's not the most sophisticated, but it actually requires about the least amount of hands-on time and plasticware of all of the ones we've tried. So until we find something better, we're sticking with it because we can still get through almost 100 samples a day. For our spike, we use an engineered lentivirus, so an envelope virus. I picked this because I wanted to add something where I knew the sequence was coming from me and not from the wastewater. Tried other things, but nothing seems to be a perfect spike. Nothing seems to exactly reproduce recovery of SARS-CoV-2. We use Chia-amp viral RNA extraction, same as what a lot of health labs use. And we're still doing qPCR. We've thought about going to digital, but every time we do the math, we find that it would take longer and cost more. For the qPCR, we still do a standard curve on every plate, which is kind of a pain, but it still gets the job done. So the one thing that really bugged me in the beginning is that when we would measure actually our recovery of SARS-CoV-2 versus our spike, they never matched quite well. In general, the SARS-CoV-2 always recovered a little bit better. And I always suspected that it wasn't really envelope particles we were seeing. I thought maybe it was ribonuclear protein complexes or something. So we actually did some tests to see if this was true. But we found that if you add detergent to your wastewater, the SARS-CoV-2 signal completely goes away, which is consistent with it being in a virus that has a lipid bilayer. And so then we finally just separated them out on a density gradient to see what density the signal is coming from. And sure enough, it had exactly the same density as our lentiviral particles. So we're almost certain what we're detecting really is viral particles that have been shed into the wastewater. However, despite this fact there's still envelope particles, we have tried many times without any success to recover any infectious virus from the wastewater. So even we've been sampling a lot of prisons, and when there's a prison outbreak, the SARS-CoV-2 level gets into the tens of millions in copies per liter. But even with those samples, concentrated or unconcentrated, we've never been able to recover an infectious particle from wastewater. One of the things we've been working on is finding a good tracer to reflect how much human contribution there is to the wastewater. Like a lot of groups, we continue to use PMMOV, although we don't think it really works very well. Actually, what we found currently is our best candidate is caffeine. Measuring caffeine is a pretty good indicator of the human contribution. So these are the sites we're sampling in Missouri. It's about 100 municipal cities and 36 state facilities that we sample twice a week. That includes all of the prisons, state mental health hospitals, and some other spots. We're pretty much covering all of the state. I don't know exactly the percent. It's over 70% of the population. But if you look at where there are people in Missouri, we've pretty much got it all covered. It's to the point where, and many of you probably heard that CDC is looking for more municipalities to participate, and they want to find places that are not already being sampled. And we're kind of having a hard time finding places that we already aren't doing. So we got a pretty good coverage. We have a dashboard. Anyone in the state can look up to see how how numbers look in their facility. It usually takes us a day or two to get the website updated from when we get the samples in the door. And if you click on any one of the cities, you can look at not only where you are today, but sort of your trends over the last year. So you can see with any one of these cities, the first wave, the second wave, the Delta wave. So you can see where you are in perspective. So the rest of the talk is going to be about sequencing. We started to look into sequencing of the SARS-CoV-2 genome from wastewater about last December, right around the time the first variants were appearing. When we were deciding what approach to use, I was kind of looking at other coronaviruses and what their evolution looks like in terms of what is the most informative part of the genome. And what you find with other human coronaviruses is that, well, first of all, almost all of the neutralizing antibodies target this receptor binding domain of the spike protein. And that is very clearly the region that evolves the fastest. And this is the region that, dictates whether neutralizing antibodies are going to change. So if you look at like 229E, another coronavirus, over time, this RBD adapts to the immune response of the human population. So that's where we really decided to target. So we mostly use targeted sequencing. These are the three sites that the three amplicons we go after. These are the three sort of information-rich regions. We worked out a software platform for analyzing data, basically removing any chimeras that come from PCR. The nice thing is we do the sequencing with a MiSeq paired end read. So we get about 550 nucleotides of continuous read. So we not only see the polymorphisms, but we see which polymorphisms go together, which is huge. It allows us to actually not only look at variants of known variants, but it actually lets us sort of look at, okay, so what is new? What combinations are we seeing that are not known in patients? So this is just an example of our output, from the RBD. Like this is an example where there was a mixture of alpha and delta variant in the sewer shed. And all of this, if anyone wants to try our primers or our approach or use our software, it's all available. It's been published a little earlier this year. So once we started doing this sequencing, we put up a second dashboard, which is our variant tracker. There's not too many of these around. This was our very first date. This was beginning of February, or in February. And when we first started, it was the perfect time to start because it was kind of boring at that point in time. All of these brown squares just means that there was no variance. It was just the original 2019 strain. There was, on this first date, there was only one location where there was any significant amount of a variant, and that was the first appearance of the alpha variant or the UK variant, B117. Over the next month, we just gradually saw alpha, spread throughout the state and displace the 2019 lineage. And it did so in a pretty predictable route. It sort of first filled out all of the sewer sheds around St. Louis, and then it sort of followed the populations. It followed the interstates out to the rest of the state. These yellow triangles means that there were multiple variants detected. So all of these, I can tell you, were alpha plus something else. We've seen just about all of the variants of concern by now. I think there's only 2 Greek letters that I haven't seen from the wastewater at this point. By the end of April, the alpha variant had mostly taken over, although we would still see some of the other variants of concern, like the California variant and some of mu and some others, but it was mostly alpha, and we basically weren't seeing any of the 2019 lineage anymore. It seems to have been fully displaced. But at this point in time, the cases were going down. Sewer shed numbers, we were starting to get non-detects for the first time since we had started, and we were debating whether we were going to continue doing our testing much longer until May 10th, when the very first time we detected this sequence called B617.2. At that point in time, it was not the Delta variant. The Greek letters had not been assigned yet, and it was not a variant of concern yet, but the World Health Organization had said that this is the fastest of the India variant. So we started paying attention to this pretty much from the beginning. We first saw it on May 10th in this town called Branson, which is a tourist destination. They have a lot of country western type entertainment, but a lot of people go to Branson and then go back home. And that might explain what we saw next, which is by the very next week, we started to see Delta appear, not in the cities, but in these rural parts of the state. And so by the next week, we saw it in this place called Lynn County, which, if you were following back in May, this was the epicenter for the very beginning of the Delta variant in the US. They had about 3 or 4% of their population infected over like a 3 or 4 week period. It was pretty bad. And within 14 days of when we first detected Delta, it was just all over the state. But again, it wasn't so much in the cities or the populous areas, but we first saw it in the rural areas. But then as time went on, by June, it was the only thing we were detecting. It was in every sewer shed and taking over the state. And our dashboard is really boring now because it's just Delta, all Delta in every location. One of the interesting things we could do from the data, though, is we could look back at first introduction of the alpha variant. What we saw is almost inevitably when alpha arrived, it would gradually displace the parent lineage. It would be gone within a matter of weeks, but it didn't have an affiliated spike in numbers. But whenever we saw the Delta variant, you could almost start a stopwatch. It was within a week to 2 weeks, there would be a giant spike in the RNA levels in the sewer shed. And then, of course, it always had corresponding patient data that eventually came through. This is one of the unexpected uses of our data. We were just talking about this earlier, that most states won't really release detailed data about what variants of concern are in what communities. But with wastewater, it's anonymized, so we can publish that away. And this was really greatly appreciated, I think, by the health affiliates, that they could just look on our dashboard and see what variants are in their community. But this reporter from Time Magazine reached out to us because they wanted to sort of have some kind of graphic to show what happens when Delta arrives. So she took all of the counties where we were doing sewer shed testing in Missouri and aligned them based on when Delta was first appeared. So time 0 here was when we first saw Delta in each of those locations. And then she plotted the publicly available patient data for all those counties. And it really showed this very clear trend that it didn't matter whatever else. Once Delta arrived, there was a corresponding increase in patients. So now I'm going to move on to the very unusual part of our work, which is this started when we first started doing sequencing. There was this one particular sewer shed in Missouri that started off with just the 2019 lineage. In February, it had a mixture of alpha and the parent lineage. But then starting in March, it had this unusual lineage that we didn't really know what it was. It had L452R, so we thought it might be the California variant, but it had this other mutation, which was N460K, which is not a very common mutation you'll see in patients. And then when we came back the next week, that lineage was still there, but it had changed. And what really freaked me out is that this N460K, it was now present twice. So there were 2 different nucleotide changes causing the same amino acid change, which is rare in and of itself, along with these other mutations that look very suspicious. This is backwards. No, that's right. 417 and 493R. And then we continued to follow, and throughout the month of March, this lineage, whatever it was slowly taking over the sewer shed. It was in some weeks the dominant sequence we would see, and almost every week it seemed to pick up another polymorphism. So all these other polymorphisms were always appearing in this backbone that had L452R. We never saw them in alpha or the 2019 lineage. But as fast as they appeared, they started to go away. So starting in April, the frequency of these sequences started to go away, and by May, they were gone, and we never saw them again. And to this date, if you look on GISAID, there is no patient or any other sequence that is even close to what we were seeing from the sewer shed. So we don't know where these were coming from. But during this time, I started reaching out to just about everyone I could think of across the country that was doing sewer shed testing. I probably reached out to a lot of people in this audience asking if they had seen anything similar to this. And most people said, we don't know. Most groups are doing whole genome sequencing where everything is broken up into small pieces. So if there is a new lineage, it's 5% of the sequence, you wouldn't really know because you can't tell which polymorphisms go together. The one exception was with New York City, with my excellent colleagues that I met after they did a podcast called This Week in Virology, and I reached out to them, and they're using a similar approach to what I was, and they also thought they had been seeing lineages that don't fit, but we didn't. They had a slightly different approach to doing sequencing than we did, so we started collaborating, and they started sending me sequences from the 14 sewer sheds from New York City. And sure enough, their sequences, they had some oddball lineages as well. They were not the same lineages we were seeing in Missouri. They were clearly different, but they had commonalities in terms of where you were seeing mutations. But what really got our attention is, well, first of all, these are very far from anything you had seen in patients, but every one of the lineages from New York City had a mutation at position 498. In particular, a lot of them had 498Y, and you actually need 2 nucleotide changes to get from Q to Y. And if you look in GISAID, there is not a single patient in the world that has ever had this mutation. However, this mutation has been seen in bats, in pangolins, and in rodents, SARS-CoV-2 lineages that infected those. And that was like a light went off. It's like, okay, maybe we're looking in the wrong place. Maybe these sequences aren't coming from humans at all. Another thing I just wanted to point out is that we see, in some cases, a whole lot of amino acid changes. This one from Wastewater Treatment Facility 3 had, I think, 16 changes, including a complete deletion of position 484, which many of you know is a really critical amino acid. And there are very rarely any silent mutations, which suggests that whatever is happening, there is a very strong positive selection that these viruses are undergoing. So after a year of doing this, I finally got to do a virology experiment again. So we generated the sequence to a lot of these receptor binding domains, and we put them into a SARS-CoV-2 spike so we can make pseudoparticles to sort of test their characteristics. And one thing we found is that they indeed, all of these New York City lineages have gained the ability to infect or to utilize both mouse and rat ACE2. Which does not mean that's where they came from, but is at least consistent with the hypothesis, because the 2019 lineage cannot utilize mouse or rat ACE2. So we've been on a hunt for what seems like a few decades now. It's only been maybe 6 months, but trying to figure out where these lineages are coming from. And I'll just tell you now, we don't know yet. We have our suspicions, but we don't know. One of the things we started doing is sequencing the ribosomal RNA from these sewer sheds to see what animals are contributing or what are the potential sources of the signal. For the most part, we pulled out what you would expect to find, which is we pull out human ribosomal RNA, but we also pull out a lot of ribosomal RNAs from things that humans eat. So we see a lot of cow, pig, goat, chicken. I don't think they're wandering the streets of Manhattan. I assume this is from consumption, but the 3 things that we frequently see that are not hopefully consumed by humans are rat, cat, and dog. So we think at this point that there is either some very odd cohort of humans, perhaps there's only GI infections or something so that they're being missed by nasal swabs, or it's coming from an animal host. And personally, I really think it's an animal host, because the one thing with the possibility that is coming from humans is we can't understand why the lineages would remain geographically constrained. Generally, the sequence we pick up from a particular wastewater treatment facility, we have picked it up consistently from that facility since the beginning of January until last week. But if it were in humans and it were spreading, you would think that it would, make its way to other places. But that seems consistent to me with an animal that is geographically constrained. So there was a paper, this is sort of making sense of what those mutations are. These graphs are from Jesse Bloom's lab. So what he has done is he made every possible amino acid change in the receptor binding domain of SARS-CoV-2 spike, and he's tested to see which ones are still functional and which mutations lead to resistance to neutralizing monoclonal antibodies. So most neutralizing antibodies bind to the receptor binding domain, and they fit into 3 or 4 classes. And within a class, they bind in a particular way. So an amino acid change that leads resistance to one class will likely lead to resistance to lots of monoclonal antibodies in that class. But when I started to go through his papers, it was just like hand in glove. It fit so well because essentially every amino acid change we have pulled out of these lineages from New York City and from Missouri are known antibody escape mutants. So wherever these viruses are coming from, they seem to be running away from some kind of immune response very fast. We also generated particles and tested ourselves whether they really are resistant to these neutralizing antibodies, and it's exactly as their genotype would predict. So like for all 4 of the New York mutants, for example, this is one of the class 1 neutralizing antibodies. This is actually one of the antibodies used in the Eli Lilly monoclonal therapy. So if you can see that in purple, this is the parent lineage. If you add more and more antibody, it goes away. Am I at 20 minutes?
Unfortunately, yes.
Okay, I'm going to skip ahead. And we have looked at every SRA in the database. The only place where we find these sequences are from New York City. From what we can gather from the sequence, these are probably something that branched off very early, but they are very clearly SARS-CoV-2. And I'll just stop there.
Thank you very much. That was a really informative, excellent presentation, and I definitely wanted to hear more. So I'm sorry your time ran out. We have a lot of questions, so I'm going to go ahead and for those of you who haven't already, please put your questions in the Q&A. So the first question is, how far apart are the polymorphisms you see? And could you check for specific SNPs that cause increased binding to the mouse ACE in other wastewater sequence data? I think you sort of addressed this after the question was asked, but I'll. Yeah,
so there's a 2-part thing there. First, the virus has to exist, and then it has to have access to the sewer shed. And most sewer sheds, there's a lot of cities on the East Coast where there's still a lot of mixed sewer sheds where stormwater and sewage mix, and then you're likely to pick them up. But a lot of places where it's closed sewage, it doesn't mean they're not there or in the animals there, but it might not be in the sewage system where we would detect it. But of all of the whatever hundreds of places that BioBot has sequenced, pretty much the only place we saw it was from New York. And we have not picked them up again in any of our 130 Missouri sewer sheds. Well, actually, no, that's not true. There was one other that had it, but then it went away. There currently are none from any of the Missouri sewer sheds either.
Are you able to submit the sequencing data that your lab generates to NCBI because of, or not?
We submitted it about 4 months ago, everything.
Okay.
So that there is a preprint. I can put that in the chat with all of this information, and all of our sequencing information is publicly available. All of the raw SRAs.
Are you capturing any animals in the catchment to sort of look for these coronaviruses that you're detecting in wastewater that might be from animals?
Not me personally, but we are collaborating with groups that are, yes, but no smoking gun yet.
Okay.
I was really thought that if this is really in rats, that we could catch a rat anywhere in New York City and they would have neutralizing antibodies, but I can tell you that that is not the case. And we've also, I can't tell you how many little packages of rat turds that my colleagues in New York have sent me, and nothing so far.
Great package to receive in the mail.
They stink so bad.
How does the wastewater data on variants compare with the clinical data?
Very well. That's actually in the preprint where we sort of look at least over the if we sort of combine all 14 of the sewer sheds and sort of make a composite of how much we are seeing of each variance and compared them with what you're seeing based on the clinical sequencing, at least based on the spring, it matched up very well. So like from January to April, we saw this big increase in alpha along with a decline in the amount of the 2019 lineage. It got a little dicey there late in the spring because you can only sequence what you can amplify. And once it gets below a certain threshold, we just couldn't amplify it. So that kind of broke, although now it matches perfect. Everyone just sees Delta.
Yeah. Okay, so I have one more technical question and then some more general ones. So this question is the variant detection only based on the mutations at the RDB? Also, are the mutation frequency always high in the sample? And is that taken into consideration?
So we have done some whole genome sequencing, and we've looked at RBD, NTD, S1, S2, and we've also done some larger PCRs. For most of the ones I've talked about, we actually have all of S1 now, and yeah, there's mutations across the S gene. The frequency varies. I suspect that the variant frequency actually is pretty constant, but depending upon how much is coming from patients, it can make it a lower or higher percent of what we're seeing. But there have been many times when it's been the only thing we detect was the oddball lineages.
Do you have a sense whether shedding of patients or infected individuals with different variants is different? Do you have a sense of that at all from your experience?
I haven't looked at it in detail, but sort of when I look at it, basically from Missouri, for instance, when the Delta variant was looking, I would look at sort of our patient data roughly relative to how high our RNA spikes were, and I didn't see a difference. It seemed pretty similar to me.
Okay. Are any of I know Missouri has a lot of rural areas. Are people on septic, and how does that affect your ability to use wastewater for surveillance in that state?
Yeah, that is an issue. We don't do septic systems. We do some small towns, and mostly we just try to get sewer sheds as diverse areas across the state as we can. So there are some counties where it'll be a fairly small town we're looking at, but it's the biggest town in the county. So I guess the reasoning would be that if they're infecting people in the really rural parts, they're getting it somewhere. But yeah, I mean, we're never going to get 100% of the state, but we are well over 70%, I believe, in terms of total population.
Great. Okay, one last question, and there's a lot more questions, but we just can't get to them all. And this question is just about sort of the issue we were talking about before the webinar, and you mentioned it about like making these data publicly available. Are there, do you feel like you have full support of like the state government and public health in releasing these data, or was there any hesitancy or pushback from anyone about like the variants and revealing information about how like different cities had different progressions or different vaccination rates and that sort of thing?
There was some hesitancy in the beginning, and we still don't release data on anything smaller than a wastewater treatment facility. So we don't release the data on prisons or state hospitals or anything like that. Anything where it's a small enough population where you could potentially track it back to a person, we don't. I mean, this was new, so there was hesitancy with just about everything, but this became such a useful tool that for health departments that they could, or it's something that, I mean, I've seen this like on web pages from health departments at counties. They'll actually point to our wastewater data as a way of convincing people, guys, look, you need to go get tested because this isn't just the flu that's circulating. See, look, here it is. Here's a graph from your waste. It's real. And it is totally anonymized. There's no way you could track it back to a person. So at least in Missouri, they have been totally on board with sharing the community level data. the community level sort of wastewater data. They still won't release patient data to go along with it, not even to me.
All right, thank you so much, Marc, for the excellent presentation and great Q&A session. Okay, we're going to transition now to Jana Huisman, who's a postdoc at ETH Zurich. And Jana, you can go ahead and share your screen.
Can you see that? Sorry.
Yes, it looks
Okay,
great, and
perfect.
we can hear you.
Good. Okay. Thank you. Yeah, I'm very excited to present the methods that we've been developing to estimate the effective reproductive number of SARS-CoV-2 from wastewater measurements. And I must say that my approach to this topic was decidedly more from the epi side of things and infectious disease dynamics than from the wastewater side. So a lot of this work was motivated by a desire to try to help the public health interventions in Switzerland and to try to control the pandemic that we're in. And I like to make this comparison to the controllers that you might know from engineering, where you have a certain target value, namely 0 infections or the least amount of deaths or hospitalizations from the disease. And you have a way of affecting this process, namely through interventions like lockdowns or universal mask wearing, vaccinations, and so on. But as is really important with any kind of controller, you also need to know the current status of your system and how much feedback you need to give to get to that desired target level. And that's for me where all of this monitoring work that we've been doing comes in. And there's a few key indicators that are really critical to keep track of from an epidemiological point of view. On the one hand, that's the disease prevalence. So the number of people currently carrying the disease, currently infected. The disease incidence, so that's the new infections every day or week, and changes to this incidence over time. So are ever more people getting infected each day, or is the pandemic actually decreasing in size? And also here, there's a nice comparison to these PID controllers, where there's sort of a direct feedback from the daily incidence. There's a integrated feedback from the prevalence, basically corresponding to the number of infections in the past and those people that haven't recovered yet, and this derivative of this growth in incidence that you all want to take into account when planning your next interventions. Of course, if you have 1,000 people being newly infected each day, this means a different thing if you currently have completely full hospitals than if you're at the very start of your epidemic. And now the important thing when looking at a disease is that it's very hard to observe infections directly. So we have different proxies of getting at some of these underlying dynamics of the pandemic. And one is to look at confirmed cases through time, so new positive tests every day, new hospitalizations, people recently deceased with COVID, as well as all of the wastewater measurements that are a good descriptor of the number of people infected in the catchment. And the work that I've been doing for the past 1.5 years mostly targeted this last part of these different variables to track. So the changes to the incidence through time. And a very useful indicator that I'm sure you're all quite familiar with nowadays is the effective reproductive number, RE, which describes the average number of secondary infections caused by an infected individual through time. And this is a useful indicator because it has this very intuitive threshold that if RE is bigger than 1, every person that's infected with the disease will infect more than 1 person. So the epidemic will grow in size. Whereas if RE is below 1, the pandemic starts to shrink. So we really want to know what the current RE is. And this is something that we've been working on since March 2020, first for Switzerland. And then we developed this, when we developed this pipeline, we also applied it to data from 170 other countries around the world to estimate the effective reproductive number through time and communicate all these results on the dashboard that you see here. This was directly used in Switzerland to inform public health policy, even leading to a sort of infamous moment towards the end of last year, beginning of this year, where the opening and closing of certain establishments like restaurants was directly tied to our RE estimates and the ones we published on our dashboard. So what I want to present today is, first of all, how we estimate this effective reproductive number, then how we managed to extend this and apply this to wastewater-based or wastewater measurements, and lastly, how we're now even estimating variant-specific effective reproductive numbers from wastewater directly. So the idea behind the pipeline is that we have these observations of the number of people newly infected each day, and we want to get to an estimate of the effective reproductive number. And we do so in three main steps. First of all, it's important, as I mentioned also before, to remember that these case observations are a delayed observation of the underlying infection process. And the RE describes properties of that infection process. So we first need to translate back from the observations that we have to the corresponding infection timeline. We can do this using information on the delay distributions from infection to symptom onset and from symptom onset to case observation, where depending on which type of observations you use, you'll have to use a different delay distribution because it takes longer for people on average to die due to the COVID infection that they got as opposed to testing positive. So we can use a mathematical trick called a deconvolution to sort of subtract the delays that we know occur between infection and observation from the observed observations and get to an estimate for the infection incidence. This you see now in the bottom left corner of the slide, where we have this line which clearly sort of predates the observed case incidence. And it's also smoother because we need to smooth out some of the weekend effects and other observational noise to get our deconvolution to work well. So that's the first step. Then in a second step, we can take this infection incidence and put it into existing methods to estimate the effective reproductive number, because this is something that's been done also for other diseases quite frequently. And there's nice existing tools out there. EpiEstim works pretty much plug and play. The only issue is that it only gives back an estimate for the uncertainty associated with the infection process when estimating RE, as opposed to accounting also for all of the uncertainty due to the observation process, like these weekend effects and so on. Which is why we added a third component, namely that we bootstrap the observed case incidence 100 times, rerun this whole pipeline 100 times, and then combine them to get nice confidence intervals around our effective reproductive number. So we started doing this based on 3 different proxies for the infections. Like I said, the confirmed cases, the hospitalizations and deaths, with the idea that each of these will be biased by ongoing problems in different ways. So for instance, the confirmed cases might be more biased by a lack of testing capacity than hospitalizations, because people who need a hospital will be hospitalized even if it takes a bit longer to get their test results. On the other hand, all of these are still within the same health system. So if the health system as a whole is overburdened in some way or other, if we're really triaging, there's no tests available whatsoever, maybe also certain populations that do not seek out healthcare at all or cannot have access to this health system, we're missing them and we're still biased in a similar way. And you can imagine that this is exactly where wastewater comes in and can give a very nice independent proxy of this entire dynamic. So this is where we teamed up with people at EAWAG and EPFL, so the team around Tim Julian, Christoph Ort, and Tamar Kohn, who you probably all know quite well, who had been measuring basically viral concentrations in the wastewater since pretty early on in the pandemic. And we wanted to see collectively whether we could use this data, these longitudinal measurements, to estimate an effective reproductive number. And the big question there that needed to be answered is, how does this wastewater trace relate to the underlying infection incidence time series? And can we basically replace this deconvolution that we had before for the case delays by something corresponding to, or that's fit specifically to the shedding process? And in this case, what we need as a delay is the shedding load distribution. So that is how much viral particles each infected individual will shed into the sewer shed in days since they first got infected, or in days since symptom onset. And luckily for SARS-CoV-2, this is something that had been estimated. It's not great data, but at least there's some estimates available of the total viral load resulting from a single infected individual in days since symptom onset. And in our pipeline, it's important to note that what we really need is the distribution, so the temporal shedding dynamics, rather than the absolute magnitude of this shedding. So we basically normalize by that and only focus on the shedding dynamics described by the shedding load distribution. So once we have that, we can basically rerun the pipeline as I described it to you before, but instead of starting with case incidents, we start with viral loads observed in the Zurich wastewater treatment plant. We apply the deconvolution with the shedding load distribution. In our case, we assumed the one by Benefield et al. Get this estimate for the infection incidence and a corresponding estimate for the effective reproductive number. And what I would like you to see here is that actually, if you now look at the RE estimated from the wastewater data, so the green and the yellow curve describe two different gene targets, and compared to the RE that we get from the confirmed cases, they actually match up pretty well. They describe a very similar increase initially, then a decrease, which is below 1, some dynamics around 1, and then it declines again. And of course, there's still some peaks here that do not match up exactly. And now this should not be taken as a signal that the wastewater RE is somehow worse than the confirmed cases. No, they're describing perhaps even different dynamics, and it's hard to say a priori which one of these is more correct. But to make sure that this wasn't just something that worked only for Zurich, we also tested it on data from a different location and even a different wastewater matrix. So we teamed up with people around Ali Bohm and Christa Wigington, who had been measuring different locations around the Bay Area, and basically did the same thing. We, of course, needed to compare to the confirmed cases from Santa Clara County now, and use the specific delays applicable to their location, but still find a pretty nice matchup between the wastewater RE and the one that we get from the confirmed cases. And here we did an additional analysis where we compare not only to the confirmed case trace directly, but also to one where we account for periods of high test positivity. So this was described with these testing adjusted cases, where basically we see that the wastewater data matches up better with the cases where we account for low test availability. So in a way, you could argue here the wastewater trace is showing a more true result. So since we submitted this paper, we've actually extended this work also to other wastewater treatment plants around Switzerland and created a dashboard where this is continuously updated, and we estimate RE every time there's new data coming in. And for certain locations now we have more than 8 months of data and see that also over this longer time period, the general trends tend to match up pretty well, with the bigger wastewater treatment plants seeming to match a bit better than the smaller ones. But now that we have more data overall, this is something that we can truly start investigating, whether there's any sort of treatment wastewater treatment plant specific effects to the feasibility. So that's all of what I showed you so far, is using a known shedding load distribution to estimate the RE from the wastewater measurements. And yeah, one can argue, okay, is the shedding load distribution that I'm using the correct one? Maybe there's certain diseases, if one wanted to apply this to a different disease where it's not very well parameterized, well known. So we also decided to turn this question on its head and say, what if we assume that the wastewater trace and the confirmed case trace, so the estimates of the effective reproductive number from both of these sources, should match up exactly? Can we find the shedding load distribution that maximizes the fit between these two signals? So you could imagine if you use a different shedding load distribution, what you're doing is shifting this estimate further or less far back in time, and you can shift it back and forth so long until you find the place where really these two seem to line up very well. And we quantified this fit in different ways, and find some estimates for the shedding load distribution for Zurich and San Jose, which sort of match up quite well. There's some non-identifiability of shedding load distributions that have the same cumulative density function. But overall, we find that the mean of the shedding load distribution would be between 6 to 9 days, which is a bit lower than the shedding load distribution that we were using before. And now the idea is that actually, if you had data that you really trust of your catchment area for a certain period of time, both for the wastewater data and the confirmed cases, you estimate the shedding load distribution, and then basically you could use this one for the rest of time, if you will. You could have a test or a training set, and then the rest of the data sets that you use this optimal shedding load distribution for. Also, we would expect this one to be kind of universal. So it would be quite interesting to estimate it based on data from a lot of different locations and see if they all give kind of a consistent signal that manages to constrain this distribution even more. So with that, I'm almost at the end of my presentation. I just briefly wanted to tell you what we've now been doing regarding variants, also because I think it ties in nicely to what Marc was saying before. So as you mentioned, you can track the proportion of different variants in the wastewater. And this now is work done mostly by Nico Berenwinkel's group, also at the ETH in Basel, where I don't know exactly what they do. I think they use whole genome sequencing of the wastewater samples, again, collected by the team from EAVAC and EPFL. And they estimate these proportions of different variants or different mutations that are characteristic of the variants through time. And it gets these kind of plots out, as you see on the right, where it's relatively boring. You first see alpha increasing and then delta taking over at some point. And now it's also very stable for delta for a while. But what we wondered is whether we can use these proportions estimated and multiply them by the measured loads in the wastewater and get sort of variant-specific loads, which also tell us something about variant-specific REs. And that's interesting because you can use this to estimate, for instance, differences in transmission fitness, or also do predictions at let's say the regional level of how many cases there might be of a particular variant through time. So this is now very preliminary work together with a student, where we see that it roughly works. So on the left column is for alpha and the right column is for delta, top for Zurich, I'm almost done, and the bottom another location. And we see that wastewater is quite noisy, but overall the trends again of these variant-specific REs from the case data and the wastewater data seem to show some similar trends. And now we're digging in a bit more specifically as to where the differences arise. So to conclude, we developed this pipeline to estimate RE from noisy observational data, extended this to wastewater measurements, and we have a pipeline that you are very free to use also on your own wastewater data, and currently implementing some explanations which will make it easier to apply also. And yeah, this wasn't work that I did alone. Here are all the people that I would like to thank. And if you scan the QR code, you arrive at our paper where there's even more people that didn't fit on this slide.
Thank you so much. That was excellent and an excellent application of the data. So I'm going to go through some of these questions. Remember to please put your questions in the Q&A box. So I guess maybe if you could go in a little more depth. I know you said you're starting to look at wastewater specific factors with respect to how well the estimate works across these different scales. So I guess you mentioned that it seems to work better for larger plants compared to smaller. Do you have any ideas about why that might be, or what's contributing to that? Is it testing penetration, or I guess, yeah.
Okay, so I guess there's multiple factors, right? One is if you would want to compare it to case data, it helps if there's good case data available, where I guess testing penetration comes into play. But now I really meant just purely looking at the estimates that you can get only from the wastewater data without comparing it to anything else. It seems that in smaller plants, there might just be more variation from day to day, which makes it harder to pick up on general trends in RE as opposed to these smaller scale fluctuations. So that's something that we want to look into, maybe even using some simulations or so, as to what sort of the smallest you can go, where you would expect to still see sort of a meaningful signal. And then I guess there's also some other parts that have to do more with the implementation of our method and with the way we estimate the confidence intervals and things like that, which struggle a little bit more when we have lots of data points that are close to the limit of detection, for instance, or the limit of quantification. We're still figuring out a little bit how we want to take that kind of uncertainty into account. So of course, when we first did this during the fall wave last year, this wasn't so much of an issue, but then it really came up during last summer as something that we need to consider better what we want to do in those low data
on the
moments.
Uncertainty, sorry, didn't mean to cut you off. On the uncertainty part, I was kind of curious because the bands on the R estimate from the clinical are much narrower than the wastewater. And I guess you said you're accounting for kind of uncertainty in the measurements, the limits of detection. What is the factor that's contributing the most, do you think, to the uncertainty in the RE estimate? Is it fecal shedding or something like that, or is it something more kind of in the quantification part of the estimate?
So I think there's two things. One is if we look at, I don't know if I have a direct comparison. No, I don't think so. But basically, one thing is that the data is noisier than the confirmed case data that we get. And so this would be, yeah, really from a data side, I'm guessing it's both in the quantification and potentially in the fecal shedding. I think it's hard for me to say exactly where that comes from. We also had some discussions in the team if there's any way we can parameterize maybe a bit better how much uncertainty is coming from really the quantification process, and if we can take that into account better. The other thing though is that currently the confidence intervals that I'm plotting here for the wastewater data should be taken with a bit of a grain of salt, because we used mostly the same pipeline as was developed really specifically for the confirmed cases. So we have a bootstrapping mechanism that was tested extensively on the confirmed cases, and where we know, okay, it makes sense with those kind of fluctuations that we observe, but we're now going back and testing that in a bit more detail, whether those same assumptions also hold for the wastewater data. So I could imagine that these confidence intervals might still shrink, or at least get adapted a little bit more, like that's perhaps with cases, you know, okay, at high numbers, we have more uncertainty in the exact day on which we observe a certain case, but potentially given limits of detection and so on, for the wastewater data, you might not have the same scaling with high loads, for instance, that they're necessarily more variable.
Right. And then you talked about the effect of variants, but I guess one question was, have you considered kind of the effect of vaccinations on shedding or these calculations?
Yes, so we've definitely thought about that a lot, also in the context of all of our other RE estimates. Mm-hmm. Because, so essentially what happens is that the RE estimate is sort of, it doesn't care about the absolute number of infections in the infection incidence that you're looking at, but rather about changes to that. So if you're always observing, let's say 10% of your total population, then your RE is going to be accurate. So if only 10% of the actual infections get tested, your RE is going to be accurate for as long as that assumption holds. Whereas if then this fraction of the total population changes, for instance, because tests become more available, the RE will be temporarily biased because it's comparing different underlying populations, if you will. Mm-hmm. And now similarly with the vaccinations, if we're basically seeing less people being hospitalized compared to the same underlying amount of infections, because they're just less likely to progress to a severe disease, then your estimate is temporarily going to be biased, but after a certain amount of time corresponding to the serial interval, this bias will be gone and it will be correct again. So long story short, I think vaccinations have an influence as they were being rolled out, but now vaccination coverage is pretty much fixed. Basically, right. This bias should be gone. Okay. And similarly, of course, and more specifically, maybe even for the wastewater, if shedding changes because of a variant or because of the vaccination, we should be taking this into account in our shedding load distribution, but it's really hard to get that kind of information.
Right. Okay. Well, I think we're out of time, unfortunately. There were a lot more questions. I'm sorry I couldn't get through to them all. But I'm going to go ahead and volunteer you. I guess either of our speakers, if people want to contact them, I'm sure they seem very open. So thank you both. This was a really great one.
Thank you. Happy Thanksgiving.
Thank you.
Thanks.
Thanks.